Hi, I'm Associate Professor Eric Olson of Northland College, and I'm presenting on longitudinal trends and ecology of the small mammal community of the Apostle Islands National Lakeshore. My co-authors are Elise Mallinger, Julie von Stappen, and Tim Van Dielen. The Apostle Islands archipelago is perfect for studying the dynamics of mammal communities within island systems. The various shapes, sizes, and degrees of isolation of the islands allows us to test island biogeography theory. Additionally, it allows us to ask interesting ecological questions due to the unique combinations of species in relative isolation from one another. Our collaborative team has been studying the mammal community of the Apostle Islands archipelago since roughly 2014. Our work represents the most comprehensive survey of the mammal community within the archipelago to date. Initially, we started with camera trap monitoring, and since then we've expanded to include small mammal trapping. And more recently, we were able to tap into an existing partnership with the Great Lakes Inventory and Monitoring Network to access acoustic monitoring data for bats. This comprehensive data set provides us with a detailed view of the distributions and relative abundances of mammal species within the archipelago. It also provides us with opportunities to examine the temporal dynamics of these mammal communities. To give you the sense of the scale of our work, here is a map showing the distribution of our survey efforts within the National Lakeshore, which is in dark gray on this map. Our camera trapping locations are the black dots. Small mammal live trapping grids are the white circles with black dots in the middle, which I apologize are a little hard to see on this map. And then we have the locations of our acoustic monitoring devices, which are represented by stars. As you can tell, our survey efforts have been widespread throughout the National Lakeshore, with every island having been surveyed by camera traps. Only one island remaining to be live trapped for small mammals, and then acoustic monitors being placed on the vast majority of the islands. From this data, we have been able to describe the diversity of species by each group across the islands. Here, the size of a pie chart gives you a sense of the diversity of species within a group that an island might support. The larger pie charts indicate the island supports 100% of the species within that group. The smaller pie charts indicate the island supports a subset of the species within that particular group. And the slices of the pie charts each represent the relative abundance of each species on an island. So, as you can see, all islands that have been monitored with acoustic monitoring devices support the full complement of bat species found within the islands. And for the non-volant or non-bat species, we see that the larger, more connected islands support a broader suite of species of these medium and large mammals, while these smaller and more isolated islands support fewer. This data also allows us to rank the mammal community in terms of relative abundance. Here we have ranked the mammal species by relative abundance and frequency of occurrence for the entire archipelago, which is represented by the black bars, and then for the mainland, which is represented by the gray bars. And we've done this by group. So we've got A, which represents the bats, B, which represents the medium to large mammals, and then C, which represents the small mammals. You can see that for the bats, the little brown bat has the highest relative abundance rank for the bats. And for the medium to large mammals, if we exclude red squirrels from the camera trap data, black bears have the highest relative abundance rank for this group. Moving into the small mammals, we see that redback voles are clearly the most abundant and widespread small mammal of the archipelago, which is notably different than what we observe for the mainland. But we'll talk more about that later. But now that you have a decent understanding of the overall project and a quick glance at the mammal community, let's focus in on the small mammal community and dig a little bit deeper. To survey small mammals, we used live trapping grids, where we used a combination of Sherman and various sized tomahawk live traps to trap small mammals. Trap grids were established no closer than 700 meters from one another, and we attempted to locate trapping grids in uniquely different habitats. We also placed trap grids at historical small mammal trapping sites. Traps were checked at least twice a day and were baited with a mixture of foods like peanut butter, oats, apples, or sunflower seeds. We collected morphological measurements on individual animals that we trapped. We identified individuals to species and sex, and then we collected hair samples and ear-tagged individuals prior to release. Here you can see the distribution of our small mammal trapping grids by year. We increased the number of sample grids as island size increased to a maximum of 6 trap grids on an island for islands like Outer and Stockton. But even the smallest islands, we required to have a minimum of 2 small mammal live trapping grids. Based on historical data, we knew the National Lakeshore was home to a subset of the species found on the mainland, with Peromyscus species, either being the white-footed mouse or the deer mouse. Historical data was notoriously bad in terms of its ability to differentiate between the species, so we weren't certain as to which of the 2 species of Peromyscus were likely to be found on the islands before we started our survey efforts. Also, redback voles, meadow voles, red squirrels, short-tailed shrews, and masked shrews. This represented the known complement of small mammal species within the island system prior to the beginning of our survey effort. So these are the species we expected to find and plan to focus most of our analyses on.
We got snowshoe hare on Outer Island. I just ear tagged it and collected some hair samples.
We also knew that snowshoe hare were on the islands as well, and so we expected that we might capture snowshoe hare on various islands. This is a snowshoe hare from Outer Island. For small mammals, there had been a number of small mammal surveys on various islands of the archipelago from 1961 to 2004, with most of those historical surveys occurring between 1961 and the mid-1980s. This historical data allowed us to examine some of the longitudinal trends in the small mammal community by comparing the known distribution of species on the islands from the early years of the park, the 1960s to 1980s, to the more recent years of the 2000s through present day. Let's start off with some general results. To do so, let's take a closer look at that earlier graph we looked at. The graph on the left shows the relative abundance, and the graph on the right shows the frequency of occurrence or distribution of small mammals of the archipelago in the black bars in comparison to the mainland, which would be the gray bars. As mentioned earlier, we see that the Myodes gamperi, or the southern red-backed vole, is the most abundant and widely distributed small mammal of the archipelago. What is important to note, however, is how different this is in comparison to the mainland, where other species are much more abundant and widespread than they are when found within the archipelago. This suggests that red-backed voles are an important member of the small mammal community within the island system, and suggests that some factor is allowing them to maintain this kind of dominance within the islands, but not on the mainland. It's also important to note that there were a number of species that were detected only on the mainland and were absent from the islands. Those include chipmunks, meadow jumping mice, and southern flying squirrels. When we examine the effects of island size and isolation on the diversity of small mammals, we found that regardless of time period, island size was a significant factor, but isolation was not. This suggests that island size might play an important role in mediating the dynamics of the small mammal community and reinforces island biogeography theory. Now let's take a look at how the small mammal community has changed since the establishment of the National Lakeshore. This map shows changes in the distribution of specific small mammal species within the park from the 1960s and 80s to the 2000s through present. Purple represents islands where the species was detected in both time periods, and teal represents islands where the species was only more recently detected in the 2000s to present time period. Orange represents islands where the species was only detected in the 1960s and 80s time period, and yellow represents islands where the species was not detected during any survey. Myodes gapperi, or the southern redback vole in the upper left, remains present on every island surveyed. Notably, they're also present on Long, Madeline, and the mainland, which are not represented on this map. Historically, redback voles were also found on Eagle Island, but no recent surveys have been done on Eagle Island to confirm their presence. Mask shrews, or Sorex cinereus, distribution appears to increase significantly throughout the archipelago, which could suggest more recent colonization of islands in the teal coloration, or more likely these changes are due to differences in sampling methodology between the different time periods. It's also important to note that the masked shrews were also present on the mainland as well as Madeline Island during more recent surveys. When we looked at the Peromyscus species, we cannot determine the species of Peromyscus for the historical data. So we had to look more generally at Peromyscus, both the deer mouse and the white-footed mouse. With this species, this group of species, we see some recent but scattered colonizations in the Teal Islands, which we suspect is a result of Peromyscus hitchhiking on human watercraft. It's also important to note that our recent surveys have also detected the presence of Peromyscus on Long, Madeline, and the mainland. Microtus pensavonicus, or the meadow vole, represents one of the most interesting changes in the small mammal community since the establishment of the lakeshore. It appears to be blinking out from the system as it was not detected during our most recent surveys on 10 of the islands where it was previously detected. These changes are significant, and we believe it has to do with the successional changes of the vegetation of the islands and potentially the competitive exclusion by the southern redback voles within more forested habitats. Notably, meadow voles are still present on the mainland and Madeline Island as well. Because redback voles appear to be such an important small mammal species for the island system, we attempted to better understand their ecology. During our surveys, one of the things we noticed was that many of the redback voles were infected with these orange parasitic mites, mainly on their ears. Essentially, nothing is really known about these parasitic mites aside from the fact that they have been associated with lower body condition in red squirrels in nearby Ontario. The map on the left shows the distribution of redback voles with mites from a subset of our data where we recorded the presence of mites, parasitic mites. We were curious about factors that might shape the probability of mite infection within redback voles in the islands. And so we did some additional analyses to better understand what those factors are that likely influence the probability of mite infection within individuals. Our analysis looked at a number of variables, including factors like Candida U dominance within the grid site, the individual vole's sex or age class, as well as things like body condition and overall vole abundance, and some interactions between variables that we thought were biologically plausible. Of all of our covariates, the interaction between body condition and vole abundance was the most significant, which can be seen by the fact that the 95% confidence intervals of the effect don't overlap 0. Here you can better visualize the effects of the covariates where infection of parasitic mites, which is along the z-axis on the left-hand side, was associated with poor body condition, which is along the x-axis along the bottom there, and higher vole abundance, which is along the y-axis there along the side. Notably, our work can't determine if mites are causing individual voles to have poor body condition, or if the parasitic mites are just more successful at infecting individuals that are already with a lower body condition. Our work does suggest, though, that mite infection may be a density-dependent factor. We have also implemented some preliminary analyses examining a suite of habitat covariates in relation to redback vole abundances and body condition. We're also examining whether or not American marten, which are fairly abundant within the archipelago and tend to prefer redback voles as a prey item, are associated with vole abundance. Our preliminary analyses have found that at grid sites where Candida U is dominant in the understory or shrub layer, there was a greater abundance of redback voles, and redback voles had higher body condition scores. We also found that marten presence and abundance seem to be significantly correlated with U dominance and to a lesser degree with vole abundance. We hope to further refine these analyses to gain better insights into the predator-prey ecology of these two species. Our preliminary analyses do suggest, though, that Candida U and potentially other habitat covariates may play an important role in predator-prey interactions for marten and voles on the islands. The complexity of the island habitats in the shrub and understory layer is impressive. The amount of coarse woody debris, curse-worthy, especially if you're trying to walk through it. Habitat features like this are less common on the mainland, not to mention the unique role that Candida U and the high abundance of coarse woody debris likely play in terms of enhancing the subnivean habitat for redback voles and potentially marten. I suspect that these unique habitat features play an important role in shaping the island predator-prey interactions. Currently, we're working on a variety of fronts to better quantify the structural aspects of habitat. Here's an example where we're looking at using remote sensing and LiDAR data to better assess different habitat features like canopy height and potentially habitat structural complexity near the ground. But if you ask anyone who hikes off trail in the Apostle Islands as much as our research team has done, they'll tell you that it's structurally complex and unique relative to even the nearby mainland. For example, I'm going to show you a video here in the next slide of just a standard patch of ground on one of the islands, and this site doesn't even have that much coarse woody debris, but you can see the high density of shrubs and understory cover in this video. I think that will be impressive.
Finally, some open forest. Oh, so nice.
We also hope to better understand the genetic connectivity of the small mammal prey community of this archipelago. Are these small mammals venturing out onto the ice or braving the cold waters to move between the islands? These are interesting questions that we hope to answer, and stay tuned to find out the results. In closing, I would like to highlight the collaborative partnership that makes this work possible. There are people that have contributed to our work, some for hours, some for months, and some for years. This work wouldn't have been possible without that collaborative relationship between the University of Wisconsin-Madison, the Apostle Islands National Lakeshore National Park Service staff, and myself and my students at Northland College. If you're interested in obtaining any copies of the research papers or reports I referenced throughout this presentation, you can contact me at the email. I thank you for your patience, and I look forward to answering any of the questions you might have.